Okay, we start here. I start from this slide, actually, the protein synthesized in endoplasmic reticulum. So we were talking about endoplasmic reticulums of two types. One is called rough endoplasmic reticulum and smooth endoplasmic reticulum. And then we have done certain extent the smooth endoplasmic reticulum functions, and certain aspects of rough endoplasmic reticulum functions also. And then we know that proteins are synthesized in rough endoplasmic reticulum. So the rough endoplasmic reticulum either is a continuation of the nuclear envelope, okay, or else it is an invagination of the plasma membrane and then is spreaded over entire cellular volume, and it does a major function of protein synthesis, which is the one essential for most of the time the body functions, because these are enzymes, hormones, etc., etc., etc. Right, whatever it is. Now, when you come to the protein synthesized in the endoplasmic reticulum, protein synthesized on the ribosome attached to RER, right? So somebody asked me a question is in the morning, right? RER contains ribosome. Is that statement correct? Yes, that statement is correct, right? Then, and then on the other hand, ribosome attached to rough endoplasmic reticulum also correct. And then we see there are ribosomes, free ribosomes also, right? But on the other hand, what happened? Ribosomes are present in the rough endoplasmic reticulum also correct, but we can't say exclusively present in the endoplasmic reticulum, right? So the protein synthesized on ribosome attached to rough endoplasmic reticulum, right, are most of the time secreted proteins. Secreted protein, in a sense, the proteins are secreted out of the cell as enzyme, as some functional proteins, and as some hormones. So these are called secreted proteins. Or else these proteins are integral membrane proteins, right? Integral membrane proteins are the one usually span across the membrane, and then they are called transmembrane proteins, or and then they are embedded in the lipid bilayer. And then soluble proteins of ER and Golgi complex, lysosome, endosomes, and vesicles are also synthesized in the rough endoplasmic reticulum. On the other than, what happens when you come to the free ribosomes, cytosolic proteins, you can see the peripheral proteins. It is interesting, isn't it? Integral membrane proteins are synthesized in the rough endoplasmic reticulum, the ribosome attached to rough endoplasmic reticulum. On the other hand, you can see the peripheral proteins of the cell membranes are synthesized in the ribosomes, which are freely present in the cytoplasm, isn't it? And then proteins transported to nucleus, some of the proteins which are essential in the nuclear function. So those proteins to be incorporated into the peroxisome and mitochondria. So the you can see the difference. Very carefully understand this. Protein to be incorporated in the peroxisome and mitochondria is synthesized in the free ribosomes, and then proteins going to be incorporated, not incorporated, the proteins which are soluble protein. Lysosome has soluble proteins, endosome has soluble proteins, as well as Golgi complex also has soluble proteins. These proteins are synthesized in the ribosome attached to rough endoplasmic reticulum. Okay, so you see the difference. Okay, so when you look at this, when these proteins are synthesized in the ribosome, they are really packaged or packed in vesicles, and then the vesicle is what happened, transported in between two compartments, right? This picture is not very clear. I will come to that picture in another place. So what happened, these vesicles which are transporting the proteins are called transport vesicles. Understand? So these transport vesicles, right, surrounded by a protein. Understand? They are surrounded by a coating protein. The vesicles are surrounded by a coating protein. These coating proteins are COP1 and COP2. We call it as coating protein complex, we call it. Now you can see COP2 target vesicle to the Golgi. right? Transport protein from RER to Golgi apparatus. Interesting, isn't it? So this process terms as anterograde transport. So underline and keep it later on. Anterograde transport mean if the protein are transported, synthesized in the RER and transported to Golgi apparatus, then it's called anterograde transport. And then there is the COP2. COP2 is what? Coating protein 2, right? That's the target vesicle to the Golgi apparatus. And on the other hand, COP1, coating protein 1, transport protein from the cis end of the Golgi complex. We will be talking about, but the Golgi complex has two ends. One is called cis end, the other one is called trans end. Cis end of the Golgi complex back to RER. This process is called retrograde transport. So you can see sometimes proteins are modified and then transported back to RER. Clear wise. So the post-translational modification, again, you recall your central dogma now, DNA to RNA transcription, RNA to, what do you call that, protein is called translation, right? So what happened with the DNA, from the DNA, when the RNA is formed, right, that process is called transcription. There also you have post-transcriptional modification. And then what happened, once the protein is synthesized, the protein undergoes post-translational modification. So some of the post-translational modification taking place in the RER, some of the post-translational modification taking place in the Golgi. So what happened, from the RER, protein goes to Golgi for the post-translational modification. Who is involved in that? COP2 is involved, right? Once the modification is done, again from the Golgi apparatus, it goes back to, what do you call that, our RER. So that is what you call. So from RER to Golgi apparatus, the transport is called anterograde transport. Involving protein is called COP2. And then retrograde transport means from Golgi complex, and when it goes back to, what do you call that, ER, that is called retrograde transport. Anyway, right? So you can see this picture. As I mentioned earlier, right? Rough endoplasmic reticulum is a continuation of what? Continuation of what? Nuclear envelope, isn't it? So you can see the nuclear envelope, the nuclear pore is there, and then you can see this, what you call, stacked rough endoplasmic reticulum. And there are three kind of regions we can identify in a rough endoplasmic reticulum. Understand? Right? So, and part of it, what you call the smooth endoplasmic reticulum. The smooth endoplasmic reticulum doesn't have what? RER. So at the end of the day, what happened, from rough endoplasmic reticulum, proteins are synthesized, and then from the, what do you call that, vesicles area, there are tubules, cisternae, and then tubules, and then vesicle area, right? There are three regions. The proteins are packed in a small vesicle and then transported towards the, what do you call, the Golgi apparatus. You can see when you look at the Golgi apparatus, right, as I said, that is also stacked compartment. And then here you can see the stacked compartments are, we call cisternae again. And then it has two phases you have. One is called cis phase, other one is called trans phase, right? So what happened? This transported to the cis region. Understand? So receiving end of the proteins coming from RER is a cis phase of the, what do you call that, Golgi apparatus. So inside the Golgi apparatus, what happened? This protein undergo modification. The protein undergo post-translational modification. And if the protein again sent back to, what do you call that, RER also. So that is what, from cis end, it goes back to RER. So one is anterograde, and then if it goes back, this is retrograde, right? So this transport, you can see this transport is actually by COP2, and then from here to here, it is COP1 again. But now what happened? Once it is modified here, it's a secretory protein, means this protein has to go out, isn't it? So this is the cell membrane. So what happened? Again, this protein undergoes, what do you call that, post-translational modification in the Golgi apparatus, and then budding out. These proteins are packed in vesicles and then budding out from the trans phase. Budding out from the trans phase. And then from the trans phase of the Golgi apparatus, these vesicles goes to the, what do you call that? These are called secretory vesicle. And then they call them, right, fuse with the membrane, and then the content will be released. So the protein is what you call now distributed. So out of the cell where it is synthesized and then it is exported now. So the protein is tacked. Tacking of the protein means carbohydrates, moieties are added. Some of the proteolytic cleavage is taking place and everything. Now the modified protein is sent out of the cell. Okay, right. So that is what you call the process of vesicle transport. So when you look at the process of vesicle transport, right, you can see there are two compartments, right? You look at the picture in there. Look at the picture in the left hand side, right? This picture, my cursor is moving. Suppose this is one of the compartment or cisternae of what do you call that, our RER Vendapulua, right? So you can see the yellow round and the right, the blue rounds are the proteins. Understand? So some proteins are secretory proteins, soluble proteins, right? Okay. Now this yellow color is the resident protein of the donor compartment. This is the donor compartment. Yeah, tamai protein synthesis karla supply karne kana. So make a donor compartment. So donor compartment, donor organ avashyai protein. So that is the donor organ also need protein. So you can see resident protein of the donor compartment. Resident protein means එක තමන්ගේ අයිති protein එක. හරිද? ඒක yellow color එක. Then බලන්න මෙතන blue color there. Blue color is one kind of protein. And then green is one kind of protein, right? So blue color one is actually belongs to where this one, right? Soluble protein again. Blue color is soluble protein. That is, this compartment needs that soluble protein. Understand? And you have the green color one. Green color is what? Membrane protein. Understand? Okay. So membrane protein. So what will happen? These proteins are now, for example, bound to, from the donor compartment, they bind to a receptor. Bind to a receptor, right? And then what happened? Budding out from the donor compartment. And then you can see there's a formation of the vesicle. And then this vesicle has what happened? Two type of protein now. One is what? Membrane protein. Membrane protein doesn't bind to the receptor. Only that soluble protein bind the receptor. That yellow color didn't go. Why yellow color didn't go? Because that is a resident protein. Okay. So what will happen? Now this is going here. And now this vesicle fuse with the membrane of the receiving compartment, recipient compartment. And in the recipient compartment, membrane samahara protein avashya unut. I mean, if some protein is needed for the recipient compartment, what happened? That will go and embedded in the membrane. The green color will be embedded in the membrane, and the soluble protein will be released into this lumen of that recipient compartment. So you can see this is the transport, how the vesicle transport the proteins are transported from one vesicle transport, right? So now you can see in the right hand side, this is the nucleus, and then you can see what is this, our Golgi apparatus, right? Okay, so you can see what happened, whatever the RER is there, rough endoplasmic reticulum, and then proteins are synthesized in the rough endoplasmic reticulum, and then those proteins are now released in vesicles, and then this is Golgi apparatus, isn't it? Lasanai. From the Golgi apparatus, you can see in the Golgi apparatus, as I told you, the Golgi apparatus facing the rough endoplasmic reticulum is called cisterna, right? And the Golgi apparatus facing the other side, what you call membrane side, that is called transcisterna, the transcisterna we call it, right? And then some area is called medial cisterna. We call it medial cisterna. Medial cisterna is the middle part of it, and this part is what you call transcisterna, and this part is we call cisterna, and then this is all together trans-Golgi network. Okay, right? You can see now when this goes and fuse it to the Golgi apparatus in the cisterna, and then this undergo modification, And from the modification, what happened? It will go to the what side? Transcisterna area. Understand? And then they will be packaged and then in different vesicles. And then the different vesicle will go to the different site actually, right? You can see now secretory pathway. Now this is what happens. Some proteins are going to be secreted out of the cell, right? So this protein packaged in packet, what do you call that? Packed in vesicles and go to the membrane and fuse it, and then protein will be released. So this mechanism, as we done again, this is what you call exocytosis. Understand? So you can see now at the same time, right? Constitutive secretory pathway, right? That is, this is what there are two pathways we don't need to know in detail. This is one called regulatory. Here also what happened? Here what happened? The bulky, right? Bulk amount is goes there, and then here what happened? One or two protein will be transported to the. You can see that one is going incorporated in the membrane, right? Or else for some other function. This is what you call constitutive secretory pathway. This is what you call regulatory secretory pathway. A lot of molecules are synthesized and packed and sent to other target area. But here what happened? The membrane protein or a protein needed for an essential function is synthesized. That is we call constitutive secretory pathway. And then there's another one we can call it as endocytic pathway. Endocytic pathway, in a sense, for example, right? You can see some of the early endosome, we call that, for example, a membrane protein. A membrane protein can be packaged in the, what do you call that? The membrane protein is there. The green color one is the membrane protein. You can see the yellow color diamond shape is one molecule. That molecule is coming into the cell, right? So that is engulfed. That process is endocytosis, and they are packaged in a vesicle. That vesicle is called endosome. Now the lysosome goes and fuse with the endosome. That is we call late endosome. Next time we call it as endolysosome. The endolysosome got to, what will happen? That will digest the, what you call the receptor, and then this will be the protein which is going to be transported will be only in the vesicle, and then that receptor molecule will be digested. Okay, and then you can see that protein will come here, right? And then right, the protein will be released into the, what you call that, Golgi apparatus, and then it will be utilized for other purposes. So this is what you call that the vesicle transport between compartment, right? RER to Golgi apparatus and Golgi apparatus to RER, or else what happened? Protein can be coming from, so the particles can come from outside also, right? Okay, the process of vesicle transport. Anyway, now we are going to talk about that endoplasmic reticulum katakara. Now we are going to talk about the Golgi apparatus. Golgi complex, the site of modification, completion, and export of secretory proteins and glycoproteins. So the glycoproteins are mostly membrane proteins. So what is happening in Golgi apparatus? Modification of the protein, completion, that means the protein are modified and completed and exported, right? So it has characteristic structure composed of 5 to 8 flattened disc-shaped membrane defined cisternae, right? So again, this is having cisternae, right? That is having 5 to 6, not like rough endoplasmic reticulum, only few, and then they stack one on top of the other like that, and then they also have a lumen, right? Secretory proteins and glycoproteins, cell membrane protein and lysosomal proteins, and some glycolipids all pass through the Golgi structure at some point of their maturation, right? So most of the proteins, whether it is glycoprotein, whether it is cell membrane protein, or whether it is lysosomal protein, or whether it is glycolipids, all these has to go through the Golgi apparatus once. The Golgi apparatus is still structurally polarized. As I told you, there is a cis phase, and then there is a trans phase. Understand? So you can look at the structure of the Golgi apparatus in this picture, right? So you can see the cisternae, 1, 2, 3, 4, 5, 6 cisternae you have. And then there is a trans phase, and there's a cis phase. And then you can see secretory vesicles. The budding out of the newly forming vesicles are there. This is the budding out of the newly formed vesicles. Now this vesicle is already secreted. This is called secretory vesicle. Sometimes what happened, incoming vesicle also shown here. That is also come and fuse, and then materials will be released inside the lumen of the, what do you call that, Golgi apparatus. Okay, so this is the connecting between the, you can see the transport between endoplasmic reticulum and Golgi apparatus is very important in a cellular function. Okay, right. So you can see now what is happening, right? So it can be a soluble protein budding out from Golgi apparatus. Now this is called transport vehicle, right? And then inside you have the soluble proteins, right? This comes and fuse here. This is called unregulated, sorry. This is called unregulated membrane fusion. Substances are released. This is called constitutive secretion, right? Okay. And then there's another way, one is what happened, secretory vesicles, right? This is a regulated membrane fusion. Here, the membrane fusion, it is not regulated, right? Nothing interference. But here what happened, the vesicles comes and fuse with the membrane, cell membrane, and then what happened, secreted. So this is what you call the hormone secretion, regulated secretion. So the regulated secretion and then unregulated secretion. Unregulated secretion is called constitutive secretion, and then regulated secretion is called regulated secretion. Okay, right. Because when you have the regulated secretion, but why we call it regulatory secretion means, now you can see a particular hormone or something is already synthesized in RER and modified in the Golgi apparatus, and then already packaged or already packed in vesicle, secretory vesicle. But it won't be released. It won't be released. So it will be released when a ligand external stimulus. So for example, something, a ligand comes and bind to a receptor in the cell membrane. Then what happens, when the ligand come and bind to the receptor, there will be internal signal transduction. Message will be sent here. When the message sent here, this will go and fuse and then release. So most of the time the hormone release, right, due to, for example, when the blood glucose increases, insulin is secreted. So when the blood glucose increases, insulin secreted means there is a stimulus that come and bind. Already insulin will be synthesized, and then it will be in the what do you call that, vesicle, packed in the vesicle. So what will happen when a receptor comes and binds the ligand through signal transduction? The message was passed through some of the messengers. That now here this ligand is called primary messenger, and then here some of the molecules will be formed, that is called secondary. Second messengers will pass the message, and then this will come in here and then fuse. So this is what you call regulated secretion, and this is the constitutive secretion. Continuously it produces the protein and it is packaged, and then continuous secretion will be there. So this is called constitutive secretion, and this is called regulated secretion. Okay, right. So now, so when you come to the functions of Golgi apparatus, now you can see this sort of a larger version of the Golgi apparatus, right? So you can see very carefully. I wanted to explain this because you will understand very easily. Now you can see this is nuclear envelope, isn't it? This is the nucleolus, and then you can see rough endoplasmic reticulum. Right? So you can see now the ribosomes attached here. The blue color one is the protein. You can see synthesized protein is the blue color one. Is released from the ribosome into the lumen of the RER. So the ribosomes are attached together to MR. Several ribosomes attached together to the mRNA. That is what you call polyribosome. So nucleus synthesize the mRNA, right? And then tRNA, rRNA, right? Here protein is in the synthesized, and then this is what you call that rough endoplasmic reticulum. So there is a what you call the vesicle will be budding out, packing this, carrying this protein to the Golgi. So you can see translation, secretion, signal remove, beginning of the glycosylation. So what will happen? There will be glycosylation. Carbohydrate is started adding here in the RER. And then what happened? It goes to the Golgi and then fuse with the Golgi apparatus. Okay. And then cis end of the Golgi, right? When the cis end of the Golgi, it will go and what do you call that, added. And then what happened? Here undergoes post-translational modification, right? What you carbohydrates are added, phosphorylation of lysosomal glycoproteins, phosphorylation can take place, and then glycosylation can take place, sulfation can take place, everything can happen, right? So what happened? That ultimately what happened? At various levels, it can be removed. Some are removed at this level, some removed this level, some removed this level, some goes up to the trans Golgi. After that, the some are removed. Understand? So the secretory granule will go and then secretion will take place. Right? Evage, membrane proteins will be that these are the green color one is membrane protein. Membrane protein will not be secreted out. This vesicle will go and join the membrane, and the membrane, the protein, the green color one will be embedded in the membrane. Right? So you can see now, this is the process happening. At the same time, you can see from the Golgi apparatus also, vesicle can be transported back to what? RER. So one is we call anterograde transport, mean from here to here, retrograde from there to here. So there are two type of what you call that proteins, coating proteins 2 and coating proteins 1 control that anterograde and retrograde transport. Anterograde transport and anterograde transport is from RER to Golgi. Retrograde is coming from the Golgi to RER. So this is the function of Golgi apparatus. So most of the time, proteins are modified. Phosphorylation, glycosylation, sulfation, and then some of the proteolytic cleavage of some polypeptides. All these are happening in the Golgi apparatus. So you can read it and see that additional note. This is what is happening in the Golgi apparatus. Okay, so we go to the next one. The powerhouse of a cell is called mitochondria, right? So when you look at the mitochondria, is made out of what you call that two type of membrane. One is we call inner membrane, other one is called outer membrane. And then you can see what happens. The mitochondria is one of the organelle, right, where it has its own DNA, right? It has its own DNA, so it can undergo what you call that replication, right? And then the inner membrane project into the matrix. The inner membrane is called cristae, and then the matrix space, and then there is an intermembrane space between the outer mitochondrial membrane and inner mitochondrial membrane. There is a intermembrane space is also there, right? So you can see now what is happening, right? The outer membrane is there, inner membrane is there, and you can see what you call that the respiratory chain. You would have done in your A level, right? That is what you call respiratory chain or electron transport, what you call system, right? And then several proteins are in that, right? The inner membrane. Give me a minute, huh? Sorry, there was a call from Dean. Okay. So this is, can you hear me?
Yes, sir.
Yes.
Okay, so now this is the mitochondria, the mitochondrial outer membrane, and then there is a, what do you call that, transport molecule we call porin in the outer membrane, and then the lipids, enzyme for lipid metabolism, that means fat oxidation, is located in the outer membrane, and then you can see that enzyme for the oxidative metabolism for the respiratory chain, we call it, is located in the inner mitochondrial membrane. And then the ATP synthesis, there's an enzyme needed, you will study later on, that is called ATP synthetase, and that is in the inner mitochondrial membrane. Okay, right. So now we go to the function of the mitochondria. The mitochondrial, inner mitochondrial membrane folded several times. The reason for that is actually, why is that? Because to increase the surface area, right? So the number and the shape of the mitochondria is important, right? The numbers of cristae they have can differ widely from cell type to cell, right? Where the tissue having more oxidative metabolism, like heart muscle, have mitochondria, particularly large number of cristae. When you say cristae, that the internal projection, right? So that is increasing the surface area. Where you have the tissue depending on more oxidative metabolism, what happens? This what do you call that? Cristae, number of cristae is increased, right? Example is cardiac muscle, where you have a lot of, what do you call that, oxidative metabolism take place. So the number of cristae is high. Okay. Even with one type of tissue, the shape of mitochondria can vary depending on their functional state, right? And then mitochondria are mobile, and they are called plastic organelles, right? And then some of the, what do you call that, cells doesn't have mitochondria. For example, if you take red blood cells, to accommodate the hemoglobin, right, it doesn't have, what do you call that, mitochondria, right? So the most of the organelles are removed when the RBC get matured, right? Even the nucleus is not there, because to accommodate, what do you call that, hemoglobin. So in those cells like RBC, right, most of the time you don't have the oxidative metabolism. You have a glycolysis taking place, but the glycolysis like a anaerobic type of glycolysis. Okay, right. And then we go to the next one, the functions of mitochondria. The most important function of the mitochondria is to produce energy by oxidative phosphorylation. Mitochondria help the cells to maintain proper concentration of calcium ions within the compartment of the cells, right? The mitochondria also help in building certain parts of the blood and hormones like testosterone and estrogen. The liver cells, mitochondria have enzymes that detoxify ammonia, right? And then that ammonia detoxification, that means the urea synthesis is now taking place in the liver. And then here the ammonia is detoxified. And then mitochondria also plays an important role in the process of apoptosis or programmed cell death. Abnormal death of cell due to dysfunction of mitochondria can affect the function of the organ. So this slide is very important, right? So the functions of the mitochondria, right? Producing energy by oxidative phosphorylation, mitochondria help the cells maintain proper concentration of calcium ion within the compartment of the cell. Mitochondria also help in building certain parts of blood and hormones like testosterone and estrogen. The liver cells, mitochondria have enzymes that detoxify ammonia. The mitochondria also play important role in the process of apoptosis or programmed cell death. Abnormal death of cell due to dysfunction of mitochondria can affect the function of the organ. Okay, and then we come to the ribosomes. Ribosomes are the cell structure or organelle that makes protein, right? So ribosome can found floating within cytoplasm. I said free ribosomes, isn't it? Okay, and then we have the ribosomes attached to, what you call that, endoplasmic reticulum also. That is, we have already done it, right? So you can see some of the proteins are synthesized by the free ribosome, some of the proteins are synthesized by ribosome attached to endoplasmic reticulum, right? So prokaryotes have 70s ribosome respectively. So the ribosome has two subunits, right? So prokaryotic ribosome is called 70S. S is actually stand for sedimentation coefficient, right? So it has a little subunit called 30S and the bigger subunit called 50S. That 70S is not the average of both, right? So the entire ribosome is called 70S. The small subunit is called 30S and the largest subunit is called 50S. On the other hand, in a eukaryotic cell, it is an 80S ribosome. The small one is 40 and the larger one is what you call that 60S subunit. Okay, right. So this is the structure of the ribosome, right? You can see the mRNA produced in the nucleus attached to the ribosome. And then the green color one is tRNA that according to the codons present in the, what do you call that, mRNA, different type of amino acids are brought to the site of ribosome, and then the amino acids are attached to each other via the peptide bond. There's a condensation reaction is taking place, and then a polypeptide is synthesized, right? So you can see in your right hand side, you have what you call that the prokaryotic ribosome as well as eukaryotic ribosome, and then these ribosomes have RNA also, right? RNA plus proteins, right? So you can see the different RNAs present in the ribosome, the 50S and 30S, and the 30S doesn't have RNA. And then you can see in the eukaryotic ribosome, you have the protein as well as RNAs are present, right? That detail, minor detail, you don't need to know, but both ribosomes are slightly different, right? That is 70S, then this is 80S. Okay. So the functions of lysosome now. The lysosome is single membrane organelle, right? But the only organelle which is having a single membrane organelle is lysosome. So that lysosome have lytic enzymes, right? So this lytic enzyme, the type of lysosome, we have primary lysosome. If it is first formed lysosome which stored acid hydrolases in an inactive form, acid hydroplanes act on acidic medium. So the pH is approximately 5 or less than 5, 4.5. And sometime what happened, this is actually the primary lysosome is the storage compartment. That is storage the enzyme. Then we come to the secondary lysosome or heteropagosome, right? It is formed by the fusion of primary lysosome and pagosome. Right? Pagosome, in a sense, you can see when a bacteria or virus is taken by the cell, right, by a process called endocytosis, right? Actually, the process is endocytosis because there is a phagocytes do this. Phagocytes are doing this. Then this process is called phagocytosis, and then the engulfed bacteria in a vesicle or in a that vesicle is called phagosome. This phagosome then fuse with the primary lysosome and then form the secondary lysosome. That secondary lysosome is known as heteropagosome, right? Okay, right. And then when we go to the types of lysosome, right? So the lysosome contain undigested material which may be eliminated by exocytosis. That is what we call residual body, we call it, right? And the same time, autophagosome, lysosome containing cell organelles, right? After digestion, right? These are called autophagosome. Understand? So mitochondria or portions of the ER to be digested, right? That is what you call that autophagosome. And then the mitochondria or any of the cell organelles is destroyed by lysosome. That process is called autophagy. The process is called autophagy. Okay, right. Now this is what I want to show you. Now you can see a bacteria is coming in. When a bacteria is coming in, right, or other way around, we take it, right? So for example, some food particles is taken. The process is called endocytosis, number 1. That is what you call, then it is in a vesicle, that is called early endosome. And then early endosome will become late endosome. And then that is what fused with the primary lysosome. Then that will become secondary lysosome, right? And then you can see, then what happened? The digestion will take place, and then a food particle will be digested. It's one process. Other than that, there's another thing happening. It's called phagocytosis. Phagocytosis, you can see, a bacterium will be engulfed, right, by the cell. That is, the process is called phagocytosis, and then what is formed is called phagosome. Now, phagosome, the primary lysosome comes and fuse with phagosome, and then you get secondary lysosome. You get secondary lysosome, right? And then from there, what happened? The bacteria will be destroyed. At the same time, a mitochondria is now old, isn't it? The no more that mitochondria should be kept inside the cell. So what happened? Around the mitochondria, the ER will form a vesicle, right? So it is the mitochondria, the old mitochondria or immature mitochondria, sorry, that might mean the unwanted mitochondria will be surrounded by what you call that ER. Then for that, our lysosome will, primary lysosome will come and fuse. And now this is called autophagosome, right? Now this is phagosome. Then it is a bacteria surrounded by the membrane, and then that is what you call that phagosome. But when a mitochondria is surrounded by ER, that is called autophagosome. And then primary lysosome will come and bind with the autophagosome, and then you get the secondary lysosome. Okay, and then digestion will be take place, and mitochondria will be destroyed by the hydrolytic enzyme. Okay, fine.
Sir, if ER is free living,
Hello.
so can the ER just no, ER is like attached to the like nucleus? No? Can it just go and wind around the mitochondria?
Yeah, it can. There is what you call the endoplasmic reticulum. When it is there, there can be. I mean, there are small. Sometimes the endoplasmic reticulum part can be freely available also, not necessarily everything attached together. Understand? It's a network of filaments, isn't it? Network of tube-like structure, isn't it? So there is a ER, which is there. This is a higher regulation. I am not going into detail, but it is engulfed by the ER only whenever it is needed to get rid of a what do you call that mature organelle. Okay. Right.
Thank you.
And so this is the functions of lysosome. Heterophagy means digestion of foreign particle through phagocytosis, very called cell eating, we call it. If it is water, then it's called cell drinking, pinocytosis. Autophagy means digestion of an intracellular substance. It can be an organelle or it can be an intracellular substance. And then lysosomes of bone eating cell dissolve the undesired part of the bone. That is what we call done by the osteoblast. That is called extracellular digestion. Autolysis. Old cells and infected cells are destroyed by this process. All the lysosome of cell burst out and then digest the cell. That is called autolysis. And then crenophagy means convert inactive hormones into active hormones through lysosome is called crenophagy. Example is thyroxine, right? And metamorphosis is during metamorphosis, the cathepsin enzyme digests tail of the tadpole of larva. That is not necessary for us. And then mitosis, lysosome are required to initiate the mitosis. So these are the functions of what we call that lysosomes. Okay. And then when we come to this is the different phagocytosis and autophagy. The same thing is explained in a different picture now. And then we go to the next one, the enzyme present in the lysosome, right? The enzyme present in the lysosome are phosphatases, right? And phosphatases, nucleases, proteases, polysaccharidases, and then glycosaminoglycans, hydrolyzing enzyme, lipid hydrolyzing enzyme, etc. And then these enzymes are most of the time they are lytic enzyme or hydrolytic enzyme, we call it, right? So the lysosomal enzyme dysfunction, any of this dysfunction will lead to a disease called lysosomal storage disease, right? There are enzymes present inside the lysosome compartment, phosphatases, nucleases, proteases, polysaccharidases, oligosaccharidases, and then all these enzymes, right, does the lytic function, right? But what happens if any of these enzymes defective? So that disease end up in a disease we call lysosomal storage disease. There is a defect called I-cell defect, deficiency of an enzyme N-acetyl glucosamine phosphotransferase. If that is there, then we call I-cell defect, right? Then there is another disease called Pompe disease. The Pompe disease is where the absence of alpha glucosidase enzyme, you end up in Pompe disease, right? So these are the diseases due to the defective, what do you call that, lysozyme enzyme. Okay, so these are the various what do you call that functions of lysosome: gene regulation, metabolic signaling, apoptotic cell death, plasma membrane repair, tumor invasion and metastasis, cell migration, cell addition, detoxification, cholesterol transport, cytotoxic killing, antigen presentation, microbial killing. All these degradation, they can be biosynthetic, endocytic, phagocytic, autophagic. All these are important functions of what you call that lysosome. Okay, right. And then there is another one, is we call it called peroxisome. That is also membrane bound organelles in the cytoplasm of almost all eukaryotes, right? So these peroxisomes, usually there are certain metabolic processes are taking place in the peroxisome, right? So these fatty acid oxidation, phospholipid biosynthesis, and then glyoxylate clearing, catabolism of purine, polyamines, and prostaglandin are taking place in the peroxisome. Is a lipid bilayer membrane surround which regulates the enters and exit of enters and exit the peroxisome. There's a membrane surrounding the peroxisome that control the substances entering and exiting the peroxisome. Okay, right. Thank you. Next one is what do you call that? The peroxisome contain several oxidases, right? So this is what you call that. Sometime what happened, there is in the oxidizing, the enzyme that use molecular oxygen to oxidize organic substances. In the process, what happened, you can get hydrogen peroxide. This hydrogen peroxide, right, is very, what do you call that, very toxic, isn't it? It can oxidize the cell membrane, protein, and lipids, and then can damage the cell membrane, especially RBC. What happens, you get damage of RBC, you get hemolysis, right? So there is an enzyme present in the peroxisome. That enzyme is called catalase. Catalase, right, remove hydrogen peroxide, right, as H2O plus oxygen. So the toxicity of hydrogen peroxide won't be there in the presence of what you call that peroxisome, right? That is essential to prevent the cell from this oxidation by these free radicals like hydrogen peroxide. Okay, right. And then come to the nucleus, right? That is the major, largest, what you call organelle present in the cell, right? And then you can see it is components of nuclei is a nuclear envelope, and then inside you have the nuclear matrix and nuclear lamina, and then nucleoplasm, and then you have nucleolus, and then there's a chromatin, and then the what you call extension of the nuclear membrane or nuclear envelope is what you call that endoplasmic reticulum, and that is consists of what you call that lot of ribosome on it, right? So when you come to the functions, is controls center of the cell. Actually, this is the that is stores and protect the information, genetic information, that's the DNA, and then it makes RNA and protein, and then makes ribosome also, right? So you can see that the nuclear pore, right, is not just a pore. It is surrounded by what you call that protein, right? So it has double membrane. One is outer membrane, other one is we call inner membrane. Okay, so this is what you call our nucleus, and then you can read up more on the nucleus. In the anatomy, people will be doing a lot on the nucleus also. And then thank you very much for this listening this lecture. It's very heavy run. Everybody understood this?
Sir.
Okay.
Yes, sir.
Yes, sir.
Yes, sir.
Right, you have to read it, and then now we are going to do the other lecture for at least half an hour. We start it, and then we can do it again. Rest in the Friday also. Shall I start the cytoskeleton?
Okay, sir.
Okay, sir. Sir?
Yeah.
Sir, I have a question regarding the Golgi complex. In the Golgi complex, all the cisternae, they are not interconnected, right?
Yes, not interconnected.
So when moving from one cistern to the other, they have to move through transport vesicles there as well, right?
Yeah. Of course, they are to move the transport vesicles. That's
Okay.
why the step by step I showed you.
Yeah. Okay, sir. Thank you, sir.
Right, very good question. Okay. Right. We are going into the section called cytoskeleton. Right. So cytoskeleton, right, as like our skeletal system in the human body, each and every cell have a cytoskeleton system, isn't it? So when you look at the cytoskeleton system, right, now this is the reference you have to do. Then I will give you the reference book later on. We send it to your email. I will get it from the batch rep. Okay, right? And then what is cytoskeleton? Somebody ask you what is cytoskeleton? Cytoskeleton is a network of filaments and tubes. We call tubules that extend through the cytoplasm from the nucleus to the plasma membrane of from the nucleus to the plasma membrane of an eukaryotic cell. So it is from the nucleus to the plasma membrane and extensively distributed inside the cytoplasm, right? And then this is a scaffolding system, supportive scaffolding system. When you say scaffolding system means, now you can see when you build up a what do you call that very highest high rising condos or something like that, they will make what do you call them scaffold, right, with iron bars and then what do you call it, bamboos, everything පාවිච්චි කරලා මේ scaffold එක හදලා ඒ scaffold එකෙන් උඩින් තමයි උඩට යන්නේ. So that kind of supportive system is given to the cell by the cytoskeleton, right? So cytoskeleton is very dynamic. It's not a passive framework. So it is always a dynamic situation, right? And the filaments of the cytoskeleton undergoes what do you call that constant rearrangement. Now you can see this. The one side of the membrane, plasma membrane, is written as PM. And then you can see the ER is there. And you can see the mitochondria is there. And then you can see other cellular organelles are there. And then you can see in that inside, you can see some of the, what do you call that, cytoskeletal element. Right? Because the cytoskeletal element is like a scaffold. You can see when you have a 5-story building, in between 2 floors, we may have a scaffold. It's like that. Right? So the blue color one is we call microtubules. And then that is involving organize the cytoplasm and often interacting with organelles. And then produce the organelles move along these microtubules. It's like highways. Organelles such as endoplasmic reticulum, mitochondria reside within the cytoskeleton framework. Angular, it holding the organelles. Understand? Okay. So when I look at the skeletal, there are three kinds of filaments inside the cell. These filaments are called microfilaments. Microfilaments, another name for the microfilaments is called actin filaments. And then another filament is called intermediate filaments. Other one is called microtubule. So when you look at the major cytoskeletal components, right? So there are three filamental system is there. So when you have a immunofluorescence micrograph, we call it. When you take the cytoskeletons can be like this. Microtubules in green color, actin will be the red color, the intermediate filaments are the yellow color. That is an immunofluorescence micrograph, we call it. At the same time, when you stain it, there is a protein stain called Komashi blue. The Komashi staining, the appearance of the cytoskeleton inside the cell will be like that. The darker color is blue color is nucleus. Other one is what you call the cytoskeletal elements. So, what is the cell? What are the cellular function of cytoskeleton? It maintains and changes the shell shape, right? Supports the fragile plasma membrane, right? Plasma membrane is fragile, right? As I told you, human body has a shape because of our skeletal system, isn't it? Otherwise, we are a bunch of muscles, adipose tissue, etc. Right? If we want to talk somebody, the bachelor, okay, Hirun is there, so I had to hold it on the head of the Hirun and then Usala Balano and make Hirun then at the. So skeleton is giving a structure to us. Like that, the shell shape is given by cytoskeleton. And then it support the fragile plasma membrane. And then it is giving mechanical linkage that the cell can bear the stresses and strain. And then it drives and guides intracellular traffic of organelles. Organelles inside the cell, from one place to another place, it moves. But it moves along the cytoskeleton, not simply move in the cytoplasm. And then intracellular transport of material. These are the main functions of cytoskeleton. Maintaining the changes the shape, cell shape, support of fragile plasma membrane, provides mechanical linkages that let the cell bear stresses and strain, drives and guides intracellular traffic of organelles, directs intracellular transport of materials. Okay. So when you come to cellular functions of cytoskeleton, another function, you can see all these are the first slide is 5, some more here. Pulls up chromosome apart at mitosis from the central plane of the cell, either side of the two poles of the cell. The chromosomes, we call chromatids, isn't it, are pulled apart, right, by what do you call that, spindle formation. By spindle, the spindles are formed by the cytoskeleton microtubules, and then enable some cells such as sperm to swim and, right, that is what you call flagella. Sperm has a tail, that is called flagella. It is made out of what? Cytoskeleton. And then movement of fibroblast and WBC to crawl across the surfaces. I told you, white blood cells, fibroblasts, they are what do you call that? That the site of infection or inflammation, the white blood cells move towards there. Understand? Provides binding site for some RNA and proteins. And provides machinery in the muscle cell for contraction. There are two kinds of protein involved. One is called actin and myosin. Okay. Sorry. What happened? Ooh. Okay. Right. I'm sorry. Right. So that is the cellular functions of mitochondria. So now you can see cytoskeleton and shell shape. Now, for example, you can see, right? Phagocytosis. Can you see that what is happening? When you have a bacteria coming, and then you can see that the animated diagram, how this phagosome is formed, right? So these antibodies are going and binding to that blue color one is called antigen. That's a foreign body. And the purple color ones are what? Antibodies. So these antibodies are recognizing the antigen and presenting it to the phagocyte. So you can see the phagocytes are engulfing. When it's engulfed, you can see what is happening. There are green color filaments are extending. The green color filaments are actually, we call it as actin filaments, right? And then what happens once the endosome comes inside, that will fuse with lysosome, and then the process is called phagocytosis, and the bacteria will be killed by the lysosome. And then you can see platelet aggregation. You all heard about what you call platelets. It's a one type of blood cell, isn't it? When the site of bleeding, what happens? The blood is clotting. So what is happening? The platelet cells, right, recruited in that area, and then they aggregate. When they aggregate, what happens? There will be extensions coming from each platelet. You can see that the diagram, the extension coming from each platelet. That due to the actin filament, and then the platelets come together. All the platelets come together and attach to each other. That is what you call platelet aggregation, and blood clotting is stopped. Or else, when you are looking for what happens, for example, actin filaments, because actin filaments is called microfilaments, is one of the cytoskeleton. It can be in two forms. One is soluble subunit and large filamentous polymer. Right? What will happen when you have a cell, right? So you can see that the cursor, your nutrient is in this side. Understand? So what happened? These actin, filamentous actin, disassemble, and then it will become soluble actin. And then again, they will assemble in the site of the nutrient available. And these filamentous actin engulf the nutrient and then take the nutrient inside, like endocytosis. So this is where cytoskeleton and cell shape change example, right? And then you can see structure and support. Now look at the epithelial cell. When you look at the epithelial cell, you can see this is having the microvilli, we call it, the projection, isn't it? The number one, where you have, what do you call that? A cytoskeleton is there. That is the structure and support. Actin filaments. The red color ones are called actin filaments. Each and every microvilli, the projections have actin filaments. And then you can see beneath that actin filaments, there are blue color filaments are there. That is called intermediate filaments. That will again give this number one. The function is number one, meaning structure and support. And then you can see light blue color one. The light blue color one, you can see this is Golgi apparatus. From the Golgi apparatus, the vesicles are going to the cell surface. It is not just going through the cytoplasm. That means the vesicles are transported along the microtubules. So the microtubules act as what? Highway inside the cell. I call it as highway. You can see to and fro, the substances are transported along the light blue color one. This is what you call, right? So these are what you call microtubule. So they are involving, you can see the organelle transport, and then vesicles carrying proteins they can transport, or else they can transport some of the nutrients from here to here. That is involvement of what you call microtubule, right? So there are, this is microtubule means there are some motor proteins involved. Again, this is the vesicle. The yellow color one is vesicle. There are two motor protein attached to this vesicle, and they walk along the microtubule and then go to the cell surface. That is what you call second one, intracellular transport. And third one is contractility and motility. Contractility and motility, third one is usually what happens, these nerve cells, right? Contractile, for example, actin filaments and myosin filaments in muscle cell. Muscle contraction, these two involve actin and myosin. Actin is a microfilament. You can see this is the neuron, and this is what you call that our axon. The axon, you can see the light blue color one is what? Microtubule. The microtubule, again, what happened? You can see the vesicles are transported. The neurotransmitters are transported from the site of synthesis to the nerve end. So you can see here, the light blue color one is called microtubule. The dark blue color ones are what? Intermediate filaments. And then you come to the dividing cell. When you come to dividing cells, actin filaments, right? After, what do you call that, cell division, chromosomes are separated, new nucleus is formed. So there should be a cytoplasmic division, isn't it? That means from one cell you have to have two cells, right? So that division is taken by the actin filaments. Cellular division in a mitosis or meiosis is due to actin filaments. And then you can see that blue color ones are called microtubule or spindles. Those are the one carrying what? Chromosome to the either side of the two pole of the cell, right? Okay. So you can see now, microtubules are tubule-like structure, right? Hollow tubes. That is 25 nanometer width. The largest diameter is associated with microtubules. And then these are actually there are some monomers, basic protein are polymerized to form the microtubule. The protein subunit is called tubulin. There are two types of tubulin, alpha tubulin and beta tubulin, right? The main functions are given: maintenance of shell shape, cell motility, chromosome movement in cell division. And then you can see microfilaments. Microfilaments, the diameter, there are two intertwined strands of actin. Always two strands, they intertwine together. That is called actin filaments. Both are actin filaments. And then you can see that they are the smallest diameter, 7 nanometer diameter. The monomeric protein is actin. The major function, maintenance of shell shape, changes in cell shape, muscle contraction, cytoplasmic streaming, cell motility, right? We call it as pseudopodia, and cell division, cleavage, furrow formation when the cell divides into two cells. That is by microfilament. Then comes the intermediate filaments. So the diameter is in between two. Microtubule is the largest diameter, and microfilament is the smallest diameter, and intermediate filament is in between that. The protein, what do you call that, the subunit, the protein subunit is called keratin. So it's a polymer of keratin. The polymer of actin is microfilament. The polymer of tubulin is what? Microtubule. Like what? The polymer of keratin is what? Intermediate filament. Right? Again, this is giving a tensile tension bearing elements, maintenance of cell shape, anchorage of nucleus and certain other organelles in the cell, formation of nuclear lamina by these proteins. Okay, so we first talk about the intermediate filaments. Intermediate filaments are two types. It can be cytoplasmic and nuclear. Cytoplasmic intermediate filaments are several types of proteins. Are the unit, monomeric unit, keratins in the epithelial surface. And there is another type of protein called vimentin. Usually you find in connective tissue, muscle cell and neurological cells. And there is another one is called neurofilaments. That is you find in nerve cells. Inside the nucleus, you have another protein called nuclear lamins. In all animal cells, you have the nuclear lamins. So intermediate filaments are two types, cytoplasmic filaments and nuclear filaments. And cytoplasmic filaments can be divided into keratins, vimentin, neurofilaments. Keratins are associated with epithelia. Vimentins are usually present in connective tissue, and the neurofilaments are usually present in the nerve cells. So you can see what is happening. You can see a cell is there, right? Okay. In your left-hand side, the cells having intermediate filaments. And in your right-hand side, the cells without intermediate filaments. So when you are stretching, what happens? The cell can rupture when it don't have the intermediate filaments. So the intermediate filaments provide the mechanical strength to the epithelial, muscle, and connective and nerve tissue. You can see in the left-hand side, when the stretching state taking place, the cells are remain intact together. Understand? Clear?
Sir,
Right.
sir.
Yeah.
Sir, that means that all the intermediate filaments are not made of keratin.
Not all the intermediate filaments are not made out of keratin. There are different intermediate filaments, and I told you, right? That is what I showed you here. Keratins in epithelia, and then in connective tissue, the intermediate filaments are formed from vimentin, right? And then in the nerve cells, they are called as neurofilament, right? And then lamins, there are protein called nuclear lamins that form the intermediate filaments in the nucleus, right? Not from keratin always, right? Anyway, but we are talking most about the keratin only. You can see now. So intermediate filaments are larger than the actin microfilaments. So the largest one is microtubules. The smallest diameter is microfilaments, and the intermediate one is what do you call it? Intermediate filaments. Okay, sorry. So that all intermediate filament proteins have this same general structure, and you will study in the protein that there are two types of protein structure we have that you have done in your A level also. One is called alpha helical structure, and then beta pleated structure. So alpha helical portion interrupted by non-helical structure, we call it. I will show you. So the most conspicuous intermediate filament, most prominent intermediate filament is keratin. There are two types of keratin we have. One is called type 1, other one is called type 2. Type 1 is acidic type, type 2 is basic type. Now somebody asked a question, then this is the answer. The major type of intermediate filament proteins. The epithelial cells, the hair and the nail, the protein is keratin. The embryonic tissue, mesenchymal cells, vimentin. Muscle tissues, right? You will study later on muscle to the Z disc, desmin, right? And then there is some cells called astrocyte, that the cells surrounding the neurons, that is called astrocytes. And then along the axons of the neuron nerve cell, there are some cells called Schwann cells. They are glial fibrillary acidic proteins. Neurons of the peripheral nervous system, the protein is called periperin. Neurons of the central nervous system, the protein is called internexin. Neurons of the CNS and PNS, neurofilament proteins, sometimes nucleus in all nucleated cells. All nucleated cells, right? Lamine. We call nucleus lamine, we call it. Okay. So when you look at the structural organization of the intermediate filaments, right? You can see it's a monomer. So that's a polypeptide chain. If you know your basic knowledge of the polypeptide chain that you have done in your A level, it has N-terminal and C-terminal. N-terminal, you have the amino group. C-terminal, you have the CWH group. And then what happened? You have an alpha helical structure. It's coiled around the imaginary axis. That is what we call alpha helical structure. And then what happened? Two monomers combine together form a dimer. Okay. And the two dimers staggered and form a tetramer. Tetramer, that means it's a staggered tetramer. Not exactly the N-terminal with the N-terminal or C-terminal with the C-terminal. You can see they are in a staggered manner, right? Forming the tetramer. You can see the first one is N-terminal in your left-hand side, and then C-terminal in your right-hand side. And then you can see other one is N-terminal in the right-hand side, and C-terminal in your left-hand side. That is what you call staggered tetramer, we call it. That is here the one dimer, the other dimer, two coiled dimers. We call coiled dimers. First one is also coiled, second one also coiled. First one is a coiled dimer, second one also coiled dimer. So the coiled dimer and coiled dimer together form a tetramer, staggered tetramer, that is called coiled dimer. And then you can see now the tetramers packed together end to end, right? Again tetramers, two tetramers packed end to end. My goodness. And then you can see at the end of the day, monomer, dimer, tetramer, and then you can see like a tetramer packed end to end, and like a what you call that unit, a filament is formed. That filament is what you call intermediate filament. Several of that combined together, a filament, that is what you call intermediate filament. Right? So you can see when you look at the intermediate filament, it's like a sheet-like structure. 8 tetramers twisted into a rope-like filament. Right? This is 8. There's 8 is there, but I don't want to show the 8 count. 1, 2, 3, 4, 5, 6, 7, 8. Right? 8 tetramers, right? Side by side and then head to tail like that, arranged, and then they, what do you call that? Rope-like filament, coiled together, and then rope-like filament. Here you have atactiano, right? This is 1, 2, the round shape. Make a balando. Ayo. 1, 2, 3, 4, 5, 6, 7, 8. And then in one, you have a tetramer. Tetramer twisted, right? And then all these are twisted together and they form a filament. That is what you call intermediate filament. So when you have there is a defect in the intermediate filament, we'll end up in a disease called epidermolysis bullosa simplex. Right? Is genetically inherited, point mutation in keratin 14 or in keratin 5, right? Change from glutamate to lysine. The amino acid glutamate is changed by lysine at 117 position. And then you will end up in a, what we call the heterodimerization. Dimerization won't take place. Keratin heterodimerization won't take place. As a result, we get a keratin protein in the basal cells, not enough. And then small friction on the surface of your hand or somewhere, you get blisters. That you can see that, right? This disease is called epidermolysis bullosa simplex, right? Genetically inherited. Due to point mutation, the keratin heterodimerization won't take place, incomplete. As a result, this happens, right? So you can see now the various type of what you call it, epidermolysis bullosa simplex, epidermolytic hyperkeratosis. You don't need to buy hard all these names. Epidermolytic plantopalma keratoderma, right? These are the things can happen, right? Different, what do you call that, level of what do you call that, skin lesions can take place when you have keratin, defect in keratin, and there are different type of keratin can be defective, right? Keratin 9, epitomolytic plantopharma keratoma, that is the corner one, this one, right? And then you can say epidermolytic hypokeratosis, this one, mutation in keratin 1 and 10. There are various type of keratin. Keratin 5 and 14, this one, epidermolysis bullosa simplex. Okay, so you can see the conified layer, granular layer, spinous layer, basal layer. Any of these can be affected. In this epidermolysis bullosa simplex, this layer is affected. Epidermolytic hyperkeratosis, this layer is affected. And then this layer is affected. Okay, right? And then we go to the next, microtubule. As I told you, this is having the largest diameter, that is formed from tubulin protein. There are two types of proteins. We have alpha tubulin and beta tubulin, right? And then because of these tubulins as form as dimer, that dimer is called heterodimer, right? So this heterodimer, you can see there is a light yellow color and then the dark brown color. It's a dimer. There's a dimer. All these dimer together, and then you get a polymer, isn't it? So that is what? Alpha and beta tubulin. And then you get a filament structure. That filament is named protofilament. And intermediate filaments එක අපි කිව්වා 8ක් එකතු වෙලා අපිට intermediate filament එක හැදුණා නේද? Here what happened? 13 tubulin molecules embedded in the, right, a ring of 13 tubulin molecule, which is 24 nanometer in diameter, embedded in the centrosome, form the nucleus and site on which the macrotubule is built, right? So this microtubule, when it is built, there are 13 protofilaments, right? Then form the outer wall of the cylindrical microtubular structure. This is a cylindrical structure inside this core. So 13 such protofilament එකතු වෙලා, right, a tubule-like structure will be formed, right? And that is, you can see here. Now this is the protofilament, right? So this is the heterodimer. 1, 2, 3, 4, 5, 6, 7, and 13 here. There's a lumen here. One is plus n, other one is minus n. So it will grow in the direction of what you call that plus n. Understand? So this is what you call microtubule, right? So you can see now, this is the tubulin dimers, and then they form the oligomers, and then that oligomers together, they can form the minus end here, the plus end here. This is called protofilament. The several protofilaments stack together and then they form a sheet-like structure. And then the sheet-like structure will close and form a tubule-like structure. That tubule is called microtubule. And then the microtubule can elongate. This is the minus end and this is the plus end. That this can, you can see the arrow I have put it, this elongate. Understand? So this is how the microfilament is formed, or microtubule is formed. Okay. So when the microtubule is formed, that is a direction of growth. That's called assembly. Right? So there are both sides you can grow, but one, the faster growing end is the plus end. Slower growing end is the minus end. And then the growth can take place in both. The direction of growth is actually towards the plus end because the plus end growth is faster. Right? There's a GTP is involved, right? Okay. Always there's a GTP is involved, mean the hydrolysis of GTP is important, right? So there's the energy is utilized when this assembly is taking place. Okay. Right. So the microtubules form certain structure, right? Have you heard about in the interface of the cell, in the cell division, we have something called centrosome, is formed by the microtubule. During a dividing cell, the chromosome carried to the two poles of the cell by spindle. The spindle is formed from the microtubule. Some of the cells have cilia. This is called ciliated cell. The cilium is actually made out of microtubules, right? So you can see when you have a cell division, right? We have the interface, and then you can see that in the prophase, you can see that interface DNA replicates, chromosome condenses, and then the nuclear envelope disintegrate, and then you can see the formation of a spindle. The spindle is actually microtubule, and then the chromosomes are arranged in the spindle, center of the spindle, and then there is what you call that pulling the chromosome to both sides, and chromatids will separate it. That is where you can see that this microtubule involved in cell division. Okay. So there are small microtubules here. This is called astral microtubule. And there's the larger one we call kinetochore microtubule. The kinetochore microtubules, and then that is the one attached to the chromosome. And then other one is that is the microtubule closer to the, this is called polar microtubule. And the microtubule attached to the, what do you call that, chromosomes. So chromatid we call kinetochore microtubules. And then that is, this is the centromere. And there is astral microtubules. And then this is what you call polar microtubules. So when you come to this centrosome, centrosome is actually have two pairs of centrioles. The centrioles are made by microtubules, right? So that is, you don't go into detail of this, but microtubules, centrioles are made by the microtubule. So you can see now, when you have a cell division like that, right, in a cancer tumor cell divide rapidly, what we can do is we can do anti-cancer drug, we call. Understand? So all these anti-cancer drugs, we are targeting the microtubules, right? For example, colchicine, colcimide binds subunit and prevent polymerization of the tubulin. Vinblastine and vincristine bind subunit and prevent polymerization again. Nocodosol is again bind subunit and prevent polymerization. So these are microtubule targeting drugs. So when the microtubule formation is affected by these drugs, cell division won't take place and tumor growth will be stopped. Understand? And that is the one, Taxol, binds and stabilizes microtubule. And that doesn't allow the microtubule to what you call shrink, it won't, the dynamic stages, dynamic nature of the microtubules is affected by Taxol. Usually the recurrent, what you call that, ovarian cancer, the Taxol is used. Actually, Taxol is actually an anti-cancer drug, right? And then that is, this is the structure of the Taxol. And then there's a Pacific yew tree. This is the plant, they isolated this drug. Now, after that, they are synthesizing it now. Then you can see now, after Taxol's treatment, the microspindle is destroyed, and then it won't be continuous, further cell division won't take place. So Taxol is an anti-cancer drug, right? And then that is affect the microtubule organization and then preventing the cell division. Okay. And then interesting part, I will come, cilia and flagella. Cilia is made out of microtubule and flagella also made out of microtubule, right? So when you look at this, microtubules are the central structure supporting cilia and flagella, right? So you can see now, both can move the unicellular and small multicellular organism by cilia and flagella, right? So when you look at the cilia, right, the microtubules arranged like this in a cilia, right? So what happened when the flagellum is made out of microtubule, and then the flagellum works, what happened? The fluid movement will take place this direction, right? This is a wave-like movement, that flagellum. And then when you come to cilia, also made out of microtubule, that is basically sweep. It is one stroke-like action, right? And then stroke, and then slowly retrieve, and then again stroke, and then that move the water fluid in one direction, and then the animal moves. So that is one of the examples you can see. The flagellum of the sperm is made out of microtubule, right? And then the whip-like motion. Some organism has the what you call cilia. The cilia is usually we call whip-like motion, right? There is a direction of the active stroke. You can see the active stroke is faster, and then the recovery stroke is slow, and then active stroke, then recovery stroke, right? So that is what is happening in the cilia, right? So some organisms, some cells move using the flagella, some cells move using the cilia, right? So in these structures, usually 9+2 pattern we have. I'm going to go into detail. Now you can see now this is one of the another example of microtubule transport. Microtubule transport, I will teach you that tutorial day. Half an hour I will teach you and then finish it off. Today now is enough. Now almost 7 o'clock, and we will stop here. The transport function of the microtubule inside the cell, we will be doing, what do you call that, tomorrow? Sorry, Friday, isn't it? Tomorrow, Thursday, isn't it? We have next section Friday, right? Hello?
Yes, sir. Friday.
Okay, so shall we stop here and then continue on Friday?
Okay, sir.
Okay. Anything you want to ask? You have to see through the notes and understand. Okay.
Okay, sir.
Okay. Bye-bye. Take care.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you, sir.
Thank you, sir.
Thank you, sir.